Over the last decade, the gut microbial community has been implicated in a wide variety of inflammation-mediated diseases. Much of the work that has guided the progress in understanding the relationship between the gut microbial community and these phenotypes relies on a dysbiosis hypothesis. Under this hypothesis, there are normal states, healthy states, which describe the gut microbial community's relationship to the host. In these healthy states, the gut commensals are well tolerated. They perform the functions that gut commensals are supposed to perform. They give enzymatic capacity to help us digest food. They help to train our immune systems, and they displace pathogens. On the host side, because these gut microbial communities are well tolerated, the host avoids adverse and inappropriate inflammation states. On the other hand, in dysbiotic states, in states such as IBD and colorectal cancer in which there's inappropriate inflammation, there's a dysbiosis or disbalance between the host and the gut microbial community. In this state, perhaps there are pathogens that have come in and caused inflammation responses in the host, or perhaps on the host side, the immune system has become compromised or overreactive, and there is therefore an inappropriate inflammation cascade mediated by inflammation pathways. Under this dysbiosis hypothesis, we can examine the state of the microbial community in healthy people and diseased people and perform a metagenome-wide association study looking for changes in either taxa or genes or both that tell us which are the biomarkers, which members of the microbial community or which gene families in the microbial community are associated with disease. This can lead to hypothesis generation or hypotheses for which we reject the null hypothesis, and we see an association of a gene or a taxa with the disease state in the host. These hypotheses can be evaluated for mechanistic understanding of how the gut microbial community impacts the host. So one example of a study which has, I think, led to some mechanistic understanding of the way the microbial community can impact host phenotype was one of the first collaborations that I was involved with, published all the way back in 2012, getting to be a long time ago now. And this is a collaboration with Janelle Arthur, who's still here at University of North Carolina at Chapel Hill, and Christian Jobon, who's now at the University of Florida at Gainesville. And this is a study in the IL-10 double knockout mouse model. So IL-10 is an anti-inflammatory cytokine. Mice that are missing the IL-10 gene are prone to inflammation. And this is the AOM IL-10 model of colorectal cancer. So these are mice that are given tumors with a genetic background that disposes them to inflammation in the presence of the AOM carcinogen. And these mice were mono-associated in the study with one of two different bugs, with either E. coli, which we think of in this case as an invasive pathogen, or E. faecalis. And in this study, what Janelle and Christian saw was that if you have a wild-type mouse just exposed to SPF conditions, these mice do not develop inappropriate inflammation. But in the presence of the IL-10 double knockout gene, again, taking out this anti-inflammatory cytokine, mono-associating the notobiotic mice with either E. faecalis or E. coli causes a spike in inflammation in these mice. But only one of these taxa, the pathogenic E. coli, can drive this inflammation to cancer. So this is a good example of a mechanistic model where a specific interaction between a specific microbe and a host with a specific genetic background leads to disease. So if the idea of the dysbiosis hypothesis is to understand individual risk, to understand how particular microbes in relationship to a particular host can drive disease, this model is really intriguing. And since this paper has been published, I think as a field, there's been an incredible amount of progress in understanding, in mechanistic detail, how this E. coli pathogen impacts the host. So in that 2012 study, we established that it was the presence of the PKS island, this particular region of the E. coli genome, that leads to a macromolecule called colobactin, which directly damages host DNA. And since that paper has been published, we've come to understand a lot more about colobactin. The structure of colobactin has been at least partially solved. And it's been come to understood by work from Hans Cleaver's group that the colobactin itself damages the DNA in a way that generates a signature that you can actually detect relative to background in the host DNA. And by looking at the proportion of colorectal cancers that have this signature of colobactin damage, we can actually begin to make estimates of what percentage of colorectal cancers could potentially be caused by this E. coli PKS package. So in the space of about a decade, we've gone from a hypothesis of how this one particular region of the genome might impact the host to being able to describe in really kind of exquisite detail the damage that happens to the host and use that signature of the damage to estimate the fraction of colorectal cancers that may be caused by this damage. So this is encouraging progress. If we think about the dysbiosis hypothesis, right, we can really get very specific and think, okay, this one pathogen is coming in, it's damaging host DNA in this particular way that leads to this risk of cancer. And we can even estimate in an entire population what that risk is by looking at the specific mechanisms of this one particular pathogen. But as I think everyone who is looking at this video knows, it's not always this easy. So it's not always straightforward to elucidate mechanism, to go from these hypotheses about what the microbial community might be doing to a mechanistic understanding that has testable predictions that we can look at in human populations. And so to begin to introduce this idea of the complexity in the microbial community, I want to highlight another collaboration with the UNC Chapel Hill researcher. This is with Temitope Keku here also at Chapel Hill. And this analysis was performed by Rashonda Jones, who is now a postdoc in Michael Garon's group at the University of Southern California, but at the time was a graduate student in my lab, and this was part of her dissertation work. And this work that Temitope has acted as the PI for is a study of colonic diverticulosis. And colonic diverticulosis is the formation of these little pouches in the gut. This is usually an asymptomatic disease, but the presence of these pouches can be a harbinger of more serious, more symptomatic diseases. And in this case, we're looking again at a metagenome-wide association study. So these are association studies where we're looking to see if there's members of the microbial community or genes associated with the microbial community that are associated with disease. In this case, this is a 16S study. So from these biopsies, we extracted DNA. We did PCR targeting the 16S ribosomal RNA gene. We generated sequences via a Luminar sequencer. And then we classified those sequences into a counts table. So for those of you who are used to doing metagenomic association study, you're very familiar with these count tables. Right? So we have the samples along one axis of this table. We have different class taxa. Since this is a 16S experiment, we can only measure the taxa. So every time we see a particular taxa in a particular sample, we just count the number of sequences here. And then there's a whole host of bioinformatics techniques that we use on these count tables that I won't go into in detail in this talk. There's normalization procedures and there's visualization procedures. So you can think of this data set, if there's 100 taxa here, you can think of this as being a 100-dimensional data set. Right? Each one of these samples has a point in some 100-dimensional space. But of course, we as humans are not good at thinking about 100 dimensions at a time. So we use techniques such as PCOA, shown here, to compress this spreadsheet from 100 dimensions down to 2 dimensions so that we can more easily visualize it. So in these multidimensional scaling or PCA visualizations, which I think most of you looking at this video are probably used to, if dots are closer to each other, it means the samples are closer to each other, and if they're further away, the samples are less similar to each other. And in this case, the red dots here represent case, and the block dots here represent control, right? But you don't need a PhD in advanced statistics to see that the case and the control are largely overlapping. Right? So this is a result that was almost entirely negative. We had this initial hypothesis that the microbial community, right, and it makes sense, right? It makes sense to think that in guts that have these pouches, that there would be a distinct microbial community from people who didn't have these pouches in their gut, right? But that hypothesis was really not supported by the data. When we actually do the experiment with a fairly large sample size here, we see almost no difference between the case and the controls. And you might think, okay, well, this is an asymptomatic disease. Perhaps there's a reason, perhaps in a more severe disease, like colorectal cancer, as we saw before, you would expect to see larger difference between case and controls. But in fact, it is my data set, my lab at least before the current coronavirus outbreak started to shut down sequencing centers, we probably see a couple of data sets like this a month. And I would say the majority of them, if not the vast majority of them, have these negative results. The effect sizes associated with microbial communities and human phenotypes tend to be really small. And as a couple of other examples of this is a study that we recently published with Ian Carroll, also here at University of North Carolina at Chapel Hill. So this is looking at associations between psychiatric measures in 100 female subjects and the microbial communities. And these are the same PCOAs. And again, you don't need a degree in advanced statistics to see that the microbial community just doesn't seem to be responding very much, or doesn't seem to have strong associations with the psychiatric measures in this case control, in this study of about 100 people. Okay. So there's been a lot of emphasis on the gut-brain barrier. There's a lot of thinking that microbes may influence our moods. This study doesn't necessarily rule those kinds of theories out, but a hypothesis that the microbial community, at least as measured by 16S, is going to be directly associated with something like a psychiatric measure from a paper test. That hypothesis is not borne out, at least at this sample size. As another example of this is a study we performed with Richard Lasser. This is unpublished data looking at metabolic osteoarthritis phenotype, and again, doing this PCA visualization, essentially no statistically significant difference between case and control. Here's another example looking at this EOE asthma phenotype. This is with Evan Dellin, also here at the University of North Carolina at Chapel Hill. James Johnson, who's currently in my lab as a PhD student, has been involved with this analysis. And we see, again, just not any strong signal between the case and the control. And I could fill up the entire seminar time showing you these many negative associations that my lab has done over the years. These are just some of the examples that I've pulled in part because they're examples from UNC researchers. So it does seem that while there are some diseases like colorectal cancer where we are making progress in understanding mechanistic associations between a specific pathogen and disease, that effect sizes in these association studies are reliably small. And I want to spend some time thinking about why this is, because it might be that that I'm going to basically argue that this reflects biology of the microbiome and not necessarily just measurement problems that we're having in the microbiome. Right? These negative results could be because we are seeing. These negative results could be because we're performing experiments with really noisy measurement techniques, and there's a real signal there, but our sequencing technology just isn't really good enough to be able to see these differences. And I'm going to argue that in general, I think that is probably not true. That I think these effect sizes are small. We're measuring small effect sizes in these microbiome association studies because the effect sizes really are small in terms of the biology. So one reason that effect sizes are so small is because we all have our own distinct microbial communities. And those differences in individual people tend to persist even in the face of environmental perturbation. And actually, one of the most dramatic examples of how strong the individual signature of each individual has for the microbial community. So in this paper published all the way back in 2011 with Steve Zeisel, also here at UNC, at University of North Carolina, Chapel Hill, and Melanie Spencer, who's now at Atrium Health, but at the time this was part of her dissertation work when she was a PhD student in my lab. And this is a study of 15 subjects brought into a hospital environment and fed an absolutely consistent diet. So these women were brought into the hospital. This is part of Steve Zeisel's low choline experiments to look at the effects of choline on buildup of fatty liver. And for a period of about two months, the diet of these female volunteers was rigorously controlled. And during this 2-month period, we collected fecal samples and then did 16S sequencing. And this is a hierarchical clustering, a alternative visualization, although very closely related to the PCOA visualizations that I've been showing you. And the coloring here is by subject. And what you notice is that there is absolutely rigorously perfect clustering by subject. So if 2 points are close to each other in the tree, it means the data points are more similar. The vector of microbes in each sample is more similar. And if they're further away in the tree, they're less similar. And the coloring is by subject, and we have essentially perfect clustering by subject. And interestingly, we get essentially the same clustering whether we use sequencing, which at the time was a next generation sequencing, which back in 2011 was a relatively new technique, or whether we used an older technique called ERISA, which was looking at fluorescence based on the size of the distance between the 16S and the 23S ribosomal RNA genes. Right. So this is sort of an example. Again, one of the arguments I'm trying to make is that it's our measurement techniques are actually fairly good, right? That we're actually able to pick up biological signals. And this is an example of that, because whether you use a sequencing technique or an older non-sequencing based technology, you get the same answer, which is that the microbial community really does not shift that much even when the diet shifts. Right? So all of these subjects are brought in, they're eating their baseline diet, then they spend 2 months in the hospital, but their microbial community, the shift in the microbial community that occurs in response to diet, and there is some shift in response to diet, but the effect size of that shift is very small compared to the tendency of the microbial community in adults to stay the same over time. So this is an example. This is one of, I think, what we're trying to fight in a cross-sectional study. Because in a cross-sectional study, you're lumping people into case and control, and then you're looking for changes between case and control. But the people in each of these categories already has a very strongly distinct microbial community, and that variation, I think, can drive down the effect size in a case control study. So one way to fight this tendency of effect sizes to be small is, of course, to have a really large sample size. And an example of a study with a relatively large sample size. Now, notice in the colonic diverticulosis study that I just showed you, that also had a fairly large sample size. So having a large sample size is, of course, not a guarantee that you're going to see associations with the microbial community. But if you grant that effect sizes are small, in part because of strong individual variation in the microbial community, then large sample sizes are a necessity. As an example of a large sample size, a recently published paper with a large sample size, this is work from Katie Meyer, also here at the University of North Carolina at Chapel Hill, and Shen Sun, who is an extraordinarily talented postdoctoral fellow in my lab currently at UNC Charlotte. And this is a study of hypertension versus normal case control study as part of the Cardia study that Dr. Meyer has been involved in. And this is a PCOA ordination. Now, at first glance, it might seem like the orange and the blue dots are really close to each other, right? So the orange dots are case and the blue dots are control, right? But in fact, if you look here, there's sort of this region of the PCOA plot that is enriched in blue dots relative to the orange plot dots. And if you draw 95% confidence intervals for the for the ordination, you see there actually is a significant difference between case and control. But we needed many hundreds of subjects in order to be able to see this difference. So if we only had 50 or 100 or 150, we probably would not have seen a significant difference. So a small effect size means that a large sample size is a necessity. So part of the argument that we're trying to make this afternoon is that the small effect sizes that we see in typical metagenomic association studies are the result of small biological effect sizes. They're not necessarily just because our measurements aren't very good. And to show you really how strong our measurements can be, this is a paper that was recently published last year in Gut Microbes. And as part of this paper, we had a series of technical replicates. And so these are the same taxa through 3 different technical replicates. And the technical replicates are separate PCR reactions and separate extraction steps and separate sequencing reactions for the same sample, right? Just dividing the sample into 3. And what you can see is that within a pipeline, right, and I think this is often true, the PCR and extraction steps are highly replicable. So across these 100 samples or so, if one pipeline, if one PCR reaction and one sequencing reaction said that, for example, fusobacteria was high in one sample, in a technical replicate of the same sample, it was also very high. The problems that tend to happen in metagenomic experiments have more to do with batches. So when you look at even within the same lab, different sequencing runs or different extraction runs over time, you can get these really pronounced batch effects. And so it's sort of a typical batch effect is shown here, right? So these are 4 different batches that were run at 4 different times, as opposed to the data in the previous slide that was run at the same time, but separate PCR and separate sequencing reactions. Here, I think it was about a year passed in between this first set of batches and this next batch. And now when you do the PCA, you see there's a very strong batch effect. Right? So in general, these batch effects, when they occur, are stronger than the biological data that we're looking at. So I've shown many examples where the biological effect size was really small, and in comparison to these batch sizes that can happen, batch to batch variation that can happen when large periods of time happen between sequencing runs. But we'll say that even across these large batches, inference can survive. So this is now looking across looking at batch 1 versus 2 and batch 3 versus 4 here, right? So we see the strong batch effect. But even when we do inference and when we just do a simple t-test and take the p-value from that t-test, looking at 2 different time points from this experiment. So these time points are reflective of treatment before and after antibiotic, right? But you can see is that even though there's this large batch effect, we get the same taxa that are significantly different in this set of patients and this set of patients. So batch effects certainly add to the background noise, but if you have a reasonably strong biological signal, you can survive these batch effects. Now, these batch effects have been studied in great detail. They're through efforts such as the Microbiome Quality Control Project, that came is a paper published a few years ago now with Curtis Studenhauer as the last author. And so we know that actually when you compare different sequencing reactions from different labs, that introduces minimal amounts of variation. But protocols, especially protocols that involve differences in different protocols for DNA extraction, can have a profound impact. So these problems like batch effects, lab-to-lab variation are serious problems. But if the effect size is large enough, it's still possible to get reproducible results across studies from different labs. And as I think a particularly dramatic example of this is unpublished work from Fernaz Fulandi, who is another. I've been very lucky with the postdocs in my lab, a very talented postdoctoral fellow in my lab. And this is looking at 2 data sets, one an unpublished data set and another a published data set for bariatric surgery. This is just doing t-tests comparing before surgery and after surgery, right? And bariatric surgery has a fairly strong effect size that's associated with it. So this is a very strong perturbation that you can make on the microbial community. But what you can see in these data is that across these data sets, so these are data sets in different labs with completely different protocols, that the signal, right, the taxa that are strongly enhanced in one cohort for bariatric surgery are also enhanced for another cohort for bariatric surgery. So if there are a large number of technical artifacts that can really make microbiome studies of the microbiome more challenging. But if there's a strong biological signal, inference can survive these potential for artifacts, and we can see consistent signals across different studies. So in thinking about reasons why the effect size can be so small in case control studies, one thing that maybe we don't think about enough is that the microbiome is an environmental variable. And as such, it's going to be different in different places, in different regions of the world. And so unlike the host genome that you have for life, your microbiome is a history of where you were, where you are, and where you grew up, and all the stochastic interactions that you had with your environment. And so Tip O'Neill, who was the Speaker of the House under President Reagan, used to say that all politics is local. And I want to make an analogous argument for the microbiome, saying that the microbiome is often local as well. And to demonstrate this is a paper that now has been submitted with Penny Gordon-Larson here at UNC Chapel Hill, and again, my talented postdoctoral fellow, Shen Sun. And this is a study of over 2,000 subjects from the China Health and Nutrition Survey. And as before, for many of the examples that I've shown you, we had fecal samples from these subjects that were taken in rural and urban China as part of Dr. Gordon-Larson's part of the project that Dr. Gordon-Larson is involved with. And as before, we generate a count table. So this is a log normalized version of the count table. But if the numbers here are higher, it means that we saw more of a particular taxa in a sample, and we did PCR ordination as before. Now this PCR ordination, there's 2,000 points here, so it's actually kind of difficult to see each individual point. But we've drawn the 95% confidence interval for the 15 different provinces that these samples were taken of in China. And what you can see is that there's essentially perfect separation by province. So the microbiome in one region of China is really distinct from the microbiome in other regions of China. And if you use perm over R squared to measure the effect size, of different metadata categories, we can see that the province explains about 20% of the overall variance in this PSA ordination, and all the other variables that really the variables that you might be interested in, such as diet, weight, all of the variables that you might be interested in from an epidemiological point of view, explain much less of the variance individually than province. Interestingly, if you do the same thing for an American microbiome study, so this is an analysis that Chen performed through her pipeline on the American Gut Project, and what we see is that the state of residence actually explains more of the variance than any other piece of metadata that's part of the American Gut Project. But the difference isn't as dramatic, right? Now, this is perhaps not a fair comparison because the AMG study is a citizen science study. There might be more variability in collection techniques that could explain the smaller effect size. But it's very tempting to speculate that, I mean, for those of you who have been to China, when there when you go to different cities in China, the diets are really different. The cultural differences between different provinces, you might argue are larger than the cultural differences between different states. So one of the songwriters of Fountains of Wayne passed away, unfortunately, in this coronavirus epidemic. But there's a Fountain of Wayne song I like, in which the chorus is "New city, same stuff." Right. So if you go to a restaurant in Los Angeles and a restaurant in New York, the diet might not be that different between those two, and that's not necessarily true for differences in China. So we can't say for sure without putting all of these data in one standardized pipeline. We can't say for sure that this effect size difference is really meaningful. But it's certainly tempting to speculate that that this difference in effect size is related to broader differences in culture in China than in the U.S. Now, what's really interesting about this data set is that we can use machine learning methods to try and make predictions within and across provinces. So when you look at the various pieces of metadata within each province, you can actually get reasonable predictions, right? So if you do like a leave one out classification, and actually this is leave 1/6 of the data out and for a supervised classification model, and try and make predictions with supervised classification models, you can get reasonably good predictions within a province, right? So the microbiome the associations that do exist within each province can be used to generate machine learning models that are able to successfully make predictions. And so if you're trying to understand within a province, for example, whether somebody was in a rural or a urban region, within a province, you can use the microbiome. You can use 5/6 of the microbiome to build a statistical model and try and predict the other 1/6. And this actually works reasonably well. You get predictions that are better than the 50%, the predictions of around 0.5 areas under the curve that you would expect by chance. There's a right, if you scramble the data and try and do the same thing, you of course get numbers closer to 0.5, right? So the areas under the curve for your machine learning models are closer to 0.5 on scrambled data. But when you try and make these predictions between provinces, right? So when you try and take the data in one province and then use that data to predict who's urban versus rural in another province based on their microbiome, you don't actually do any better by chance, right? So you make your predictions of who's urban versus rural, the microbiome are indifferent when you go across provinces, right? So it's not true that there aren't differences within each province. There aren't differences within each province that you can use to classify, for example, this urban-rural difference. Right? So within each one of these provinces, you can look for urban-rural differences, and you will see significant differences. But they're not the same significant differences in each province. When you try to take one of the urban-rural differences in one province and use it to predict the urban-rural differences in another provinces, that just doesn't work at all. Right? And so this again comes back to this idea that the microbiome as an environmental variable is local, right? The way that different groups of people interact with their microbiomes are different. And so of course, if you're doing a cross-sectional study across large regions of space, this is going to impact your effect size. There's not only individual variation in the microbiome that we saw in the study with Dr. Zeisel that you have to overcome in a case control study, but there are regional differences in the microbiome. And if you're putting people together just within a single model in a case control study, those regional variations will also impact the effect size that you observe for the microbiome. And of course, it is always true that when you have unexplained variants in science, you can say, well, we're not necessarily measuring the right thing. And so most of the data that I've shown you has been from 16S rRNA experiments, right, which only is measuring one gene in a bacterial genome. And our universe kind of expands. We can get a better look at this dark matter when we start to look at whole genome metagenome shotgun sequence data. And of course, over the last 4 or 5 years, as the price of sequencing has come down, it's become more and more feasible to look at whole genome metagenome shotgun sequence data instead of just 16S. And you find really interesting things when you do this. So in an earlier study that we did with Dr. Gordon Larson in an earlier cohort from a single province within China, when we used whole genome metagenome shotgun sequence data, we saw that there were differences in not only in bacteria between rural and urban communities, but in archaea and viruses as well. And a lot of this variation is just unexplored. And so it's also true. We have a Shen Sun, a postdoctoral fellow in my lab, is working on a project right now where she's using the whole genome metagenome shotgun sequence data to assemble whole genomes from the individual reads, and what can look like a small difference in 16S ribosome RNA can actually mask differences that span hundreds of genomes when you actually reassemble these genomes, or at least tens of genomes. I shouldn't exaggerate. At least tens of genomes when you expand these genomes into whole genomes by assembling the short reads together. So we have a manuscript that Shen has just started to work on that should be coming out shortly. And so I think there's a lot of really interesting things that are going to happen in the next couple of years as we start to turn our attention from 16S to whole genome metagenome shotgun sequence data. I should say that I think 16S rRNA experiments are still incredibly useful. There are times when you can get cleaner data from 16S rRNA rather than whole genome data, and we certainly encourage our collaborators to work with both technologies. So how do we pursue, right? I hope I've convinced you that most case control studies are going to have small effect sizes. You need to have large sample sizes. You need to be able to compare across studies to understand what kind of variation is tied to a particular region and what kind of variation is consistently associated with disease. So if effect sizes are small and we need large sample sizes, we need to be able to compare across studies. And a project that my lab has been working on for a while now, which is probably the most complex thing that we have ever attempted to do, is to try and make what you can almost think of as version control for figures. So what we would like to have is an environment where, so these are all of the different steps that can happen within a typical metagenomics analysis. So you have sequences, you have many different programming languages, you have many different algorithms. As any of you who've tried to install a bioinformatics pipeline, even when you have a script that is published as part of a paper, it can be quite difficult to actually get the proper computing environment set up to run that script. So one of the goals of this project, which was started by a graduate student in my lab, Michael Ciota at UNCC, and we have a very talented analyst who's been working also on this project, Ivory Blakely, also in my lab at UNCC. So the goal of this project, which we call BiologJ, J for Java, because it lives kind of within a Java environment. The goal for this project is that I could give you the raw sequences and one file. And that one file basically specifies every single thing that happens to your sequences. And we can put this in a Docker container so that all of the software and everything that you use is already basically downloaded and installed within the virtual environment of a Docker container. And you could just execute this on whatever environment you want, whether it would be your local computer or the Amazon cloud. And it would produce the figures and tables in your paper. Right? So this way, instead of just giving someone a script and saying, well, here's the script that I ran, and maybe you'd be able to run that script, or maybe you wouldn't, or maybe the software environment that the script is running in would run properly, or maybe it wouldn't. Instead of doing that, we would have this one common language, really, that describes all the steps in the pipeline. And then a meta-analysis would be simply, if we had 100 studies like that, and in fact, we are currently undertaking a goal. We're trying to get to 50 studies. So we're trying to pick the 50 most, what we think are the 50 most important figures and tables that are in the literature. And we're trying to reproduce them. So build them all out with their own configuration file so that you could generate these 50 figures from the literature by just running these 50 scripts within this pipeline. So this is a very ambitious project. It's been far more complicated and far more difficult than I thought it would be. As many bioinformatics labs, I think, are working on these sort of core pipeline tools. They're very challenging to work on for many technical reasons that I won't get into. But we are making really, thanks to the efforts of the people in my lab, we're making some really good progress on this. And I hope to have our first publications out on this project within the next year or so. And finally, of course, it is not enough to generate mechanistic questions. It's not enough to do hypothesis generation in human studies. You want to be able to ask mechanistic studies. You want to be able to ask mechanistic questions, which requires studies in animal models. And animal models are incredibly useful, but have some really interesting challenges. And this is work that actually this week just came out, is on ISMEJ, which has been a collaboration between my lab and Ian Carroll's lab. And this is looking at a large study in which a large number of samples were put into slurries, and then the slurries themselves were sequenced, and then the slurries were put into mice. And what's interesting is if you actually look at the community composition between the fecal samples and the slurries, it was very well preserved, right? So this is again this idea that the PCR step and the extraction, the PCR step and the extraction step and sequencing steps within a pipeline generally don't introduce a lot of variance, right? So when you go from a fecal sample to a tube and resequence with a separate PCR reaction, you can see a very similar overall community composition. But when you go to the mouse, the community composition in the mouse and the human, what was in the human, can be actually quite different. Right? So there are some microbes that survive transfer to the mouse, but the microbes that survive transfer to the mice really vary substantially by taxa. So Bacteroidetes are much more likely to make a transfer between human and mouse than Firmicutes. Right? And so when you're asking mechanistic questions, this is probably something that you want to track in animal models. You want to understand what is the bug doing in the human host, and how likely is that bug to survive the transfer from the human host to the mouse host. And you want to be able to track these kinds of issues so that you can ask questions about whether the microbe can be having similar mechanistic impacts in humans and in a mouse model. And finally, of course, as a PI, I don't actually do any work myself anymore. I just try to motivate and hold together the incredibly talented people that I work with in my lab. So thanks. I've tried to highlight the contributions that people have made as we've moved through the data today. And I also, of course, have to acknowledge my many outstanding collaborators at many different institutions throughout the country and throughout the world, and our funding sources. Thank you very much.